Hello friends, this is the third lecture of our calcium metabolism series. In this lecture we are going to understand how the bone resorption and bone formation will take place. Right? So let's get started. Uh, Let us revise a bit from our previous lecture. The cell responsible for bone formation, as you guys already know, it is the osteoblast, and the cell responsible for bone resorption are the osteoclasts. Right? Now osteoblasts are basically came from the fibroblasts. So osteoblasts are the modified fibroblasts. Now how this differentiation will occur, how the fibroblasts get modified into the osteoblast. This is basically by one transcription factor known as RUNX2. Now the full form of RUNX2 is runt related transcription factor 2. Alright, let me write it down for you. It is runt related transcription factor 2. Now osteoclasts are also a modified monocyte. So monocytes somehow modified and get differentiated into the osteoclast. Now that differentiation is not as simple as the differentiation of fibroblast into the osteoblast. This one, the differentiation of monocyte into the osteoclast, is very complicated. So we are going to understand it thoroughly. Now we learn step by step. See, this is the monocyte. This monocyte is going to converted or differentiated into the osteoclast. Now this differentiation from monocyte to the osteoclast will take place with the help of another cell. That cell is either osteoblast or T lymphocyte, but most of the time it is osteoblast. Now osteoblast has a two ligand, and the same fashion monocyte has a two different type of receptor according to that ligand. Alright, now let's talk about this red ligand situated over here. It is a name of that ligand is RANKL, RANKL, and the receptor for that RANKL is RANK. So RANK is a receptor that is present over the monocyte, and RANKL is a ligand, RANK ligand, that is present over the osteoblast. Clear? Now the second one, this violet one, is the MCSF, and the receptor for MCSF is CSF1 receptor, CSF1R. Alright, now let's discuss the full form of each of them, starting with the RANKL. Now the full form of RANKL is receptor activator for nuclear factor kappa beta ligand. Let me write it down. Receptor activator for nuclear factor kappa beta ligand, and RANK is nothing but RANKL receptor, and CSF is the colony stimulating factor 1 receptor. Alright, so this MCSF is nothing but monocyte colony stimulating factor, and the receptor for it is the colony stimulating factor 1 receptor. Now here's the catch. Whenever this osteoblast or T lymphocyte get activated, this MCSF and RANKL ligand will attach to the appropriate receptor. Then this monocyte will converted into the osteoclast. So this receptor are already present over the monocyte. Once this osteoblast get stimulated, this ligand will face on the surface and will attach to the monocyte and perform this differentiation from monocyte to osteoclast. Now to inhibit this conversion, to regulate the total amount of monocyte that converted into the osteoclast, to make in check, there is one more thing that you need to remember, is this one, osteoprotegerin. Remember the term osteoprotegerin. This osteoprotegerin has also competitively inhibit with this RANK to bind it. So this osteoprotegerin, once they bind with the RANK, it will inhibit this conversion from monocyte into the osteoclast. Clear? Let me repeat one more time. we want conversion of this monocyte into the osteoclast, the multinucleated cell that is situated over the bone surface, right? Now this monocyte has a two receptor, one is rank, another one is CSF1R, and for them there is appropriate ligand that is present over the osteoblast or T lymphocyte. One is RANKL, another one is MCSF. These two things, when they bind with the appropriate receptor, this monocyte will converted into osteoclast. And now this conversion, to keep in check, there is one more thing, that osteoblast will secrete this portion, substance osteoprotegerin. This osteoprotegerin will competitively inhibit with this RANKL to bind with the rank. Now once this osteoprotegerin bind with this rank, this conversion will not take place. So this one is the inhibitory compound that inhibit the formation of new osteoclast. Now up to this discussion, I deliberately make a mistake that to make you understand that, see, this osteoblast has a only single red color ligand. That's why I make this ligand red color. This MCSF and osteoprotegerin both are secreted from this osteoblast. They are not present over the surface of the osteoblast. Got it? But to make you understand, I show you as a ligand over the osteoblast. Let me repeat myself. Osteoblast has only one ligand, RANKL, RANKL. This MCSF and osteoprotegerin both are secreted from osteoblast. They bind with the appropriate receptor to make necessary changes over the monocyte into the osteoclast. Got it? Now how to write it down? Starting with this, two distinct signaling pathway are initiated. One is the RANKL and rank interaction between the cell. In this case, RANKL is over osteoblast and rank is the receptor that is present over the monocyte. This one is the first signal. Second signal is MCSF is secreted by osteoblast and it can bind with its appropriate receptor CSF1R, means colony stimulating factor 1R receptor, and combination of these two signaling event will leads to differentiation of the monocyte into the osteoclast, right? You can also write it down over here. The osteoblast can also secrete the osteoprotegerin. That osteoprotegerin will competitively inhibit with the rank and inhibit the conversion of the monocyte into the osteoclast. This is all about the osteoclast differentiation from the monocyte. Now we already made the osteoclast. Now how this osteoclast is going to resorb the bone or erode the bone? Right, now just focus in this figure. This one is the bone surface area. This pink color cell is the osteoclast. Now this osteoclast has a attachment protein. That protein will going to attach over the bone surface. That protein is the integrin. Once the integrin will bind with the bone, it create a very private portion of that cell that is known as sealing zone. Once this sealing zone is created, two things will take place inside this sealing zone or inside this private space that is created by this osteoclast. One is displacement of this hydrogen ATPase pump. Once this hydrogen ATPase, this green colored are the hydrogen pump. Once this pump will be translocated over this surface, it secrete the hydrogen and make this sealing zone acidic. Once this sealing zone will become acidic, the lysosomal granules of this multinucleated osteoclast will disintegrate inside this sealing zone and secrete a particular substance, protease enzyme. This one is cathepsin K is the protease enzyme. Got it? So let's write it down up to that thing. Osteoclast attached to bone via integrin and create an isolated area. That isolated area is known as the sealing zone. And that sealing zone, two things will happen inside that sealing zone. Once it become pH become acidic, and second one, it secretes cathepsin. Now what this cathepsin will do? Cathepsin will break the collagen, and in some book that space, that region this private space that is created by this osteoclast, they known as Howship's lacunae or Howship's lacunae. Got it? Now here's the thing, how we are going to write it down in exam, this entire erodic how to osteoclast will erode the bone, starting with this top osteoclast attached to the bone via integrin. It will create an isolated area between the bone and osteoclast and make a sealing zone. You can also mention over how many lacunae over here, and then this hydrogen ATPase pump will be translocated over the isolated area and make the area acidic approximately up to the pH 4. Right now, acidic pH will dissolve the hydroxyapatite. There is a formation the hydroxyapatite. There is calcium, phosphate, and the hydroxyl ion. This acidic pH will dissolve this hydroxyapatite, this mineralization, and the cathepsin K, the remaining cathepsin K that is secreted by osteoclast, they break down the collagen. So basically cathepsin K is acid protease, it break down the collagen. Now we already break down the main two things of the bone. One, that is hydroxyapatite is the inorganic compound and collagen is the organic compound. Right now here's the thing, once the cathepsin K will break down the collagen, there is a breakdown product will produce. The name of that breakdown product is pyridinoline. Right, let me repeat one more time, the breakdown product, the name of that breakdown product is pyridinoline. That pyridinoline is measured in urine as an index of the rate of the bone resorption. That thing I am not mentioned in this slide, but you can write it down somewhere. The breakdown product of collagen is pyridinoline. Let me give you a spelling, pyridinoline. Right, and by breakdown of organic and inorganic product, they are forming the shallow depression in the bone. Right, now let's learn one more time. Let's have a summary of this entire discussion. Just follow my leads. These are the bone, these cells are the osteocyte, this one is the osteoclast, and this one are the pre-osteoclast, means these are the monocyte. Right, this surface small cell that is present over the bone surface are the osteoblast. Now this osteoblast has a Rankel ligand over it, and also it secretes the MCSF. Once this pre-osteoclast or monocyte will bind with the appropriate ligand, this one is the rank, this one is the rank receptor, the receptor for Rankel. This one is the receptor for MCSF. One, this rank will bind with the Rankel, and this receptor for CSF1R will bind with the MCSF. This pro-osteoclast or monocyte get converted into the osteoclast. Once this osteoclast get activated, it's bind with the appropriate portion over the bone, the area that we are going to resolve, that is known as the integrin protein. The osteoclast will bind with the bone via integrin protein and make a specific sealing zone. Once this sealing zone is made, what will happen? There is a translocation of the hydrogen pump over the margin of this sealing zone, and it will secrete the hydrogen ion and make this structure acidic, or make this lumen acidic. Once the lumen acidic become up to 4 pH, the lysosomal granule will disintegrate over here and secrete cathepsin K. Now here's the thing, this cathepsin K will break down the collagen, and this acidic pH will break down the hydroxyapatite and release the calcium and phosphate and hydroxide ion over inside the circulation. Got it? Now this osteoblast also secrete this OPG. Now what is this OPG? It is osteoprotegerin. This osteoprotegerin will bind with the same receptor and make a competitive inhibition to this Rankel and prevent the conversion of this pre-osteoclast or the monocyte into the osteoclast. Right? This is all about the summary of our discussion. Now let's discuss the bone remodeling. Now before we start our bone remodeling, let me remind you one more thing over in this diagram. See, once we want bone remodeling, we want to create this portion, this space we have to create via osteoclast, and this osteoclast, as we know, will be converted or activated from monocyte via osteoblast. So to perform bone resorption function, we want to activation of osteoblast, right? So let's discuss about the bone remodeling. As I told you in our previous lecture, the major regulator of bone remodeling is the osteocyte, the cell, the osteoblast that get entrapped inside its own secretion of extracellular matrix. Now remodeling occur along the line of the mechanical stress. Now that mechanical stress will be sensed by the osteocyte. So the osteocyte are the mechanosensor of the bone. Now it has a direct connection with the osteoblast, so it sends signals to the osteoblast, and that osteoblast will recruit the osteoclast via Rankel and MCSF, and osteoclast perform its resorption function. Now once the resorption will be performed, now new osteoblast will fill that resorbed space to form a new bone, and that new bone once formed, it will be mineralized, and osteoblast inside that will become trapped and converted into osteocytes. That's it. That's all about the bone remodeling. So for bone remodeling, we have to first activate the resorption. Once the resorptive activity will over, it make a space, then that space will be invaded by osteoblast. That osteoblast will secrete the collagen fiber and then mineralize that collagen fiber via calcium phosphate and hydroxide ion, make hydroxyapatite, and this osteoblast will be entrapped inside its own secretion and converted into the osteocyte. Now how this bone remodeling will be regulated? That bone remodeling will be regulated by a bone resorption. So once some substance will promote the bone resorption, will increase the bone remodeling. That the substance will inhibit the bone resorption, will decrease the bone remodeling, right? So the substance that are promote the remodeling, that substance that promote the bone resorption, and substance that are inhibit the bone resorption. Let's discuss, let's enumerate the first, the stimulator or the promoter that will promote the bone resorption. That's our vitamin D, parathyroid hormone related peptide, parathyroid hormone, then corticosterone, prolactin, right? So remember that vitamin D and parathyroid hormone will promote the bone resorption, and see the corticosteroid will also resorb the bone. So in corticosteroid side effect, there is a more amount of bone resorption and ultimately leads to the fracture. Clear? Now this prolactin will stimulate the bone resorption, so it will release the calcium from the bone inside the circulation, and that calcium is utilized to make a milk. Got it? Now substance that inhibit the bone resorption are the estrogen, calcitonin, calcium itself, and thrombospondin. See, the calcium itself inhibit the bone resorption, so whenever the serum calcium level is increased, it prevent bone resorption, so it prevent more amount of calcium that get released by a bone resorption. Now what is thrombospondin? You heard the terms thrombospondin. It is coming across your mind in the platelets topic. So thrombospondin is actually secreted by the platelet alpha granules. You might be remember that thing. The platelet has a two type of granule. One is alpha, another one is beta. This alpha granule will secrete this thrombospondin, and the thrombospondin causes platelet aggregation. Now there is one more term that is very similar to the thrombospondin. That term is thrombostatin. You heard that up. Thrombostatin is also again present inside the platelet. It is a contractile protein and it is responsible for clot retraction. Now how the different hormone will affect the bone resorption? Now parathyroid hormone and vitamin D, they do not have a direct receptor over the osteoclast. See, this one is the very interesting fact. Parathyroid hormone and vitamin D, both the hormone will stimulate the bone resorption, and yet both of them do not have a direct receptor over the osteoclast. So they first activate the osteoclast via osteoblast. You remember the previous figure like this. This parathyroid hormone and vitamin D both have a receptor over osteoblast. They do not have a receptor over osteoclast. So whenever the vitamin D and parathyroid hormone will increase inside the circulation, they bind with the osteoblast and will recruit the more amount of osteoclasts, convert more amount of osteoclasts from monocyte and causes bone resorption. Got it? And via that it stimulates the bone resorption. Now next one is the calcitonin. Now calcitonin is the hormone that is secreted by thyroid gland parafollicular C cell. It has a receptor over the osteoclast and it inhibits the function of osteoclast, so it inhibits the bone resorption and prevent bone resorption. Alright, we are going to discuss in our next lecture about calcitonin in detail, so don't worry about that. We are going to discuss all these three hormones in our next lecture: vitamin D, parathyroid hormone and calcitonin. Now here's the interesting thing: effect of estrogen on bone resorption. Many examiners are very much interested in the effect of estrogen on bone resorption, so I make a particular separate slide of the effect of estrogen on the bone resorption. So from the previous slide you might be remember that estrogen is going to inhibit the bone resorption. How it will inhibit the bone resorption? Let's discuss that thing. Estrogen has a receptor on both over the osteoblast and over the osteoclast, so it inhibits the bone resorption and it inhibits the release of calcium from the bone. It has a three different action. One is over osteoclast, it decreases the activity of osteoclast, it increases the activity of osteoblast, it increase the osteoblast proliferation from the fibroblast obviously. And the remaining one thing that estrogen do, it increase the synthesis of extracellular matrix. Obviously whenever the estrogen will increase the osteoblast formation, this osteoblast is going to secrete the extracellular matrix, so osteoblast via this osteoblast, sorry, the estrogen via this osteoblast will increase the synthesis of extracellular matrix, right? So this is the three primary action of the estrogen over the bone. It decrease the osteoclastic activity, it increase the osteoblast proliferation and increase synthesis of extracellular matrix. If you remember up to that thing, that's okay, but good students might also know what estrogen will do to decrease osteoclastic activity. Let me give you that detail also. It will increase the apoptosis of the osteoclast. So what is the apoptosis? Apoptosis is the programmed cell death. It causes programmed death of the osteoclast. Second thing, it increases the production of the osteoprotegerine. Now what the osteoprotegerine will do? Osteoprotegerine will competitively bind with the rank and inhibit the rankle, RANKL, and decrease the recruitment of osteoclast. These are the effect of estrogen by which it decreases the osteoclastic activity. First one is it increases the apoptosis of the already present osteoclast. Second one, it decreases the production of new osteoclast. It decreases the differentiation of monocyte into the osteoclast via increasing in the production of osteoprotegerine. That osteoprotegerine will competitively bind with the rank receptor that is present over the monocyte and decrease the conversion of that monocyte into the osteoclast and decrease recruitment of the osteoclast. Got it? That's all about our today's lecture. Now next time when we meet, we are going to discuss about the hormone that affect the calcium metabolism. Thank you.